Mikang Sim from Kongung University in South Korea. My poster title is PLR-GEN, a method for generating pseudo-long reads from metagenome short reads. Metagenomic sequences containing all sequenced genetic materials in environment samples are one of the most important resources for understanding the roles of microorganisms in an environment. In the studies using the metagenomic sequences, the creation of high-quality metagenomic assemblies is critical to accurately discover the composition and function of microbes within the environment. Recently, hybrid assemblers that use long reads together with short reads have been developed, but the generation of long reads requires relatively higher sequencing cost than short reads, thus limiting their application for a metagenomic assembly. In this study, we present a new method called PLR-GEN, which creates pseudo-long reads from metagenomic short reads. PLR-GEN generates PLR through the following 6 steps based on metagenomic short reads and reference microbial genome sequences. In the mapping and piling up step, metagenomic short reads are mapped to each reference genome separately using bowtie2. All aligned reads are filtered and piled up on each reference genome using SEM tools and pileup. Unmapped, low quality, duplicated and secondary aligned reads are discarded. In the PLR container construction step, reference genomes are broken at regions without having any mapped reads. Each of the resulting sequence fragments is defined as the PLR container, which is a template for generating PLRs. Mapped reads in each PLR container are then used to generate PLR sequences in downstream steps. In the PLR graph generation step, multiple read sequence alignment in each PLR container is converted to a graph called a PLR graph, which consists of 2 types of node and directed edges. Specifically, a normal node is created from contiguous alignment columns, where a single nucleotide occupies each alignment column. If an alignment column has 2 or more different nucleotides, then the bubble node is created for each different nucleotide. Finding combinations of bubble nodes at different alignment positions is the main problem in PLR sequence generation. For doing that, special reads called bubble linking reads that span 2 or more bubble nodes in different alignment columns are identified. The bubble linking information collected from one of the bubble linking reads is then represented as a vector of bubble nodes. In the bubble combination identification step, vectors of bubble nodes created from each bubble linking read in the previous step are used to find different combinations of all bubble nodes in the PLR graph. The basic idea is to cluster vectors of bubble nodes based on the same nucleotide in the same vector element and use each resulting cluster to define a specific combination of bubble nodes. For this purpose, a hierarchical agglomerative clustering algorithm is used. The distance can be defined only for 2 vectors that share at least one vector element containing the same or different nucleotide in each vector. Based on the calculated distances, hierarchical agglomerative clustering is carried out and the final clusters are defined using 0 as the distance cutoff. After that, the combination of bubble nodes is constructed by integrating bubble nodes at different alignment positions. When all combinations of bubble nodes are identified, final PLR paths are generated by assembling flanking normal nodes and identify the consensus bubble paths. Finally, in the PLR sequence generation step, for each PLR path, the final pseudo-long read sequence is constructed by concatenating nucleotides of normal and bubble nodes in the path. PLR-GEN was successfully applied to short read by creating PLRs with N50 of 33 kbp that could be almost completely aligned to the known reference genomes in the HMP dataset with a very low error rate. One of the excellent features of PLR-GEN is its ability to distinguish genome fragments with only a very small number of nucleotide differences from individuals of the same species or different species. For example, in this figure, two VPLRs created from the same PLR container having only 3 positions with different nucleotides were mapped to 2 different genomes of species. Finally, to examine whether PLR can improve the quality of metagenome assembly, short-read only metagenomic assembly were constructed with the metaspade. When the short-read assembly was further assembled by metaspade, the PLRs generated by PLR-GEN could reduce the number of sequences, produce longer contigs consistent with the known reference genome in the HMP dataset, and increase assembly contiguity compared with the short-read assembly. In this study, we present a new method called PLR-GEN for generating pseudo-long reads. One of the excellent features of our method is its ability to capture subtle sequence variation resulting from individual genomes in the same or different species in the metagenomic sample. PLR-GEN can be used to generate artificial long-read sequences without spending extra sequencing cost, thus aiding various studies using metagenomes. Many existing metagenomic assemblies generated by only using short reads are a good target of PLR-GEN. Thank you for listening.